Hi, my name is Felipe-Piedra. I'm an assistant professor at Baylor, and sharing our research is important. And that's why I'm here today with my new friend Hayden to talk about my work. Hello, Hayden.
Hi. So is it okay if I ask you a few questions?
Please.
So my first question is, what inspired you to start working on a solution for microplastic?
Yeah, that's a great question. And I'll start by saying it goes beyond microplastics to include macro and nanoplastics, right? So even smaller and bigger. I started doing this research through a general concern with sustainability. So you and I take part in a highly technological civilization that does a lot of things incredibly well, and it does some things poorly. And that includes the endless generation of enormous amounts of waste. So we can compare that to the way a highly evolved ecosystem works, where essentially there's no waste. Every waste product generated by an animal or plant ends up being an input into some downstream creative process. So for me, the goal is to have a research program that helps nudge our highly technological civilization more toward the way a highly evolved ecosystem works, where there is no waste.
Another question that I have is, how do the microplastics impact not only our day-to-day lives, but also our health?
Yeah, so this is answering this question involves pointing to a fairly new line of research. And there are concerning early results that micro and nanoplastics inside of our bodies might even be driving or contributed to chronic disease.
So what is PET, and how do you teach bacteria to eat PET?
Yeah, so PET is one kind of plastic, polyethylene terephthalate. The way you teach a microbe, a bacterium or a kind of fungi or any microscopic organism you can imagine to eat something like PET is sort of mysterious. Evolution involves sampling many things at once and finding solutions. So this species of bacteria that I work with that eats PET was discovered in one of 300 sediment samples taken from a plastic bottle recycling facility. We have no idea how long it took to evolve the ability to eat PET. But this idea of teaching it is very interesting. You can imagine setting up experiments where you do guide the evolution, and people do this already, where you guide the evolution of microbes to accomplish a certain task. In the case of the problem that we're considering or the research question that I'm trying to answer, you can imagine teaching this PET eating bacteria to eat it faster, right? And that essentially involves doing a bunch of experiments side by side and watching what happens, right? You need a good readout for plastic consumption, and then you select the population of microscopic organisms eating the plastic faster for further culturing, right? So you're picking the guys who do it faster, ignoring everyone else. And in principle, if you do that enough, you might end up getting a population that's very different from what you started with, and one that is much more able to consume plastic quickly.
How do you test the bacteria that you are working on?
So I test the bacteria for their ability to consume plastic by putting them in a tube. And this is actually a plastic tube, but it's a different kind. So in this other plastic tube, which I think is made out of polystyrene, I put a little liquid media that's like a culture broth that the bacteria like to grow in. And I also add a strip of the plastic that previous studies confirmed it likes to eat. And what I've been observing for 2 years now is that the bacteria will specifically grow on that piece of plastic added to the culture. They do not grow on the walls of the tube, right? Indicating that they don't want to eat that material, but they grow on the little strip. And what I can also do is measure the mass of the strip before putting it in the culture. And then I can wait a week, take it out again, clean it of bacteria, dry it out so I know I'm just looking at plastic, and weigh it again. Every time I do that, its mass decreases, right? So that's proof that the bacteria are eating that plastic specifically.
How long would you say that it would take for the PET to be consumed by bacteria?
I think that under ideal conditions, that is controlled environmental conditions where, let's say instead of fermenting hops and wheat and barley to make beer, you're fermenting plastic waste to make microbial biomass. Under those ideal conditions, if we deployed a system like that worldwide, maybe we could convert all existing PET waste into microbial biomass in 10 to 20 years.
And how would you say that this waste could be reused?
Yeah, that's also a brilliant question. Going back to the highly evolved ecosystem where there are no waste products, everything is input into a downstream creative process, you can convert plastic waste into microbial biomass. Microbial biomass is stuff that properly treated, that is, if you heat sterilize it, that is you destroy the microbes and you desiccate them, so you dry them to make a powder, that could be returned directly to soil. So you could use that to support a garden, you could use it to improve deteriorated ecosystems, especially in contaminated settings. And I'm playing more with the idea too, or exploring possibilities associated with the idea of even using dry microbial biomass to make things. So last year I had a high schooler working in my lab who was working on making recycled paper using not just recycled wood pulp, which is what paper ends up being, but also using microbial biomass derived from waste plastic. So that's one other possible use.
What other problems have you thought about researching next?
Yeah, so the lab is focused on turning PET or polyester, polyethylene terephthalate waste into microbial biomass as quickly as possible. But the next problem is to focus on other polymer types. If we just go outside and take a short walk, we're going to find PET waste, we're going to find polyethylene waste, we're going to find polystyrene waste, that's styrofoam, we'll find PVC waste. So there are other polymer types that we've released into the environment at the scale of like hundreds of millions of tons. And those all need a solution of the sort that I'm imagining, where you essentially ferment it into microbial biomass. And then another possibility is collecting all solid municipal waste and turning it essentially into one material, and then focusing on getting that consumed by microbes. Those are like the three major things. And then finally, the lab that I'm building is sort of focused on exactly that, the general idea of turning all environmental contaminants into microbial biomass. Because with that, you can build a thriving world.
So why do you enjoy doing this type of research?
Well, it brought me here, and that's pretty awesome. But the other thing is, I like contemplating things. I like stepping away and thinking through a problem. That could have led me to philosophy, which it did. But in science, you also do stuff, right? In science and engineering. And when you step away and think about the world, inevitably you see there's something about it you don't quite love, and maybe you can help fix it. So then you might be able to take an approach through science to do that. So that's partly why I like doing research. The other thing is, you can't fully explain these things. My mom is an artist and a cook, and my father's a physician scientist. All those things I feel kind of combine in what I'm doing. When you set up an experiment, it's actually not that different from doing one in your kitchen. In science, you have to be very careful, and you have to keep good track of what you're doing in order to revisit your results and really make sense of what you're doing. I believe deeply that what I'm doing matters, and that it, in some way, indirectly, it is helping me to grow all of those trees and all those happy ecosystems that I'm constantly visualizing within my mind.
Those are all the questions that I have for you today.
Thank you for your time, Hayden. It's my pleasure to be here.